Hi everyone, this is our lecture on genetic terms. So let's get started. Let's start off with a case. Let's say a 45-year-old woman presents to the clinic because her sister was recently diagnosed with breast cancer, and they both have the BRCA1 gene mutation. The woman wants to know if there is any chance that she will not develop breast or ovarian cancer. Our learning objectives for this lecture are to define genetic terms and to associate genetic terms with clinical examples. The first genetic term we are going to discuss today is codominance. Codominance is defined as having both alleles contribute to the phenotype of the heterozygote. Remember when you first started learning about genetics and alleles? Very often the example is used that the capital letter B would be dominant and stand for brown eyes, and the lowercase letter b would be recessive and stand for blue eyes. Well, what would someone's phenotype be if they were a heterozygote or had the genotype big B and little b? Well, then the dominant gene would be expressed and the person would have brown eyes. Now that's not the case in codominance. In codominance, both alleles are shown in the phenotype of the heterozygote, and some examples are the blood groups, alpha-1 antitrypsin deficiency, and the HLA groups. So let's talk about the blood groups a little bit. If your blood type is A, then can you remember what antibodies are in the plasma? Yep, you're right if you said anti-B. Can you remember what antigens are on the surface of the red blood cells? Yep, then we would have A antigen. Blood type A would have A antigen on the surface of the red blood cells. So what if you're blood type B? Well, then you'll have anti-A in the plasma and B antigen on the surface. Well, what if your blood type is AB? What antigens will you have on the surface of your red blood cells? Yep, you'll have both A antigen and B antigen. They are both contributing to the phenotype. Now, honestly, my favorite example of codominance isn't even human. It has to do with this image. Isn't it cute? This is a calico cat, and it is a white cat with black and orange patches. One of the genes that encodes fur color is on the X chromosome, and it has two allelic versions. One is for orange fur, X with a capital B, and one is for black fur, so X with a lowercase b. A cat that is heterozygous will have both colors, and the ones that have both orange and black are almost always female, because it got both X's. The next term we're going to discuss is variable expressivity. It's defined as patients who have the same genotype but have varying phenotypes. The example that you can think of is neurofibromatosis type 1. Neurofibromatosis type 1 is an autosomal dominant neurocutaneous disorder with a mutation in the NF1 tumor suppressor gene on chromosome 17. There are different presentations of the disease despite the same genotype. Some people will manifest with cafe au lait spots, like in this image. Intellectual disability, cutaneous neurofibromas, like in this image. Lish nodules or pigmented iris hamartomas, as seen here. Optic gliomas, pheochromocytomas, seizures, or focal neurologic signs. You can use the mnemonic Cyclops to help you remember all those. But these different presentations are varying phenotypes for the same genotype. Next, let's discuss incomplete penetrance. Incomplete penetrance means that not all individuals with a mutant genotype will show the mutant phenotype. We can say that it's not completely penetrable, just a certain percent. That percent penetrance multiplied by the probability of inheriting the genotype can calculate the risk of expressing the mutant phenotype. For example, just because someone inherits gene mutations in the tumor suppressor gene BRCA1 does not necessarily mean that that person will develop breast or ovarian cancer. Next up is pleiotropy. Pleiotropy means one gene contributes to multiple phenotypic effects. For example, a person with untreated PKU displays lots of characteristic findings like light skin, intellectual disability, and a musty body odor. Next, let's discuss anticipation, which means increased severity or earlier onset of disease in succeeding generations. The classic example that should come to mind is trinucleotide repeat diseases like Huntington disease. Huntington disease is an autosomal dominant trinucleotide repeat expansion in the Huntingtin HTT gene on chromosome 4. Can you remember which trinucleotide is repeated? Yep, you're right if you're saying CAG. So let's look at an autosomal dominant pedigree like this one, and let's look at the right side. So let's say this woman had 30 repeats, and the onset of her disease was at age 50. In the next generation, her daughter had 100 repeats, and the onset of her disease was at age 40. And in the generation after, this woman has 300 repeats, and the onset of her disease is at age 30. So in each succeeding generation, there is earlier onset of disease and more repeats, and possibly increased severity of the disease also. Next, we have loss of heterozygosity, which means if a patient inherits or develops a mutation in a tumor suppressor gene, the complementary allele must be deleted or mutated before cancer develops. Now, this is not true of oncogenes. It's for tumor suppressor genes. What should come to mind is retinoblastoma and the two-hit hypothesis, HNPCC or Lynch syndrome, and Li-Fraumeni syndrome. When we talk about a hereditary cancer syndrome, usually individuals are born with the so-called first hit, but don't develop cancer because there is still a good copy of the tumor suppressor gene allele to function normally. If the other allele gets hit somehow, like with a deletion or a mutation, then we can say the person has lost its heterozygosity, and more likely than not, that will lead to the development of cancer, like retinoblastoma. Next, let's talk about what a dominant negative mutation means. It means that the mutation exerts a dominant effect. So a heterozygote produces a non-functional altered protein, but this mutation prevents the normal gene product from functioning. So let's say, for example, a single mutated p53 tumor suppressor gene, it will result in a protein that is able to bind DNA and block the non-mutated p53 from binding to the promoter. The next term we have is linkage disequilibrium. It's defined as the tendency for certain alleles at two linked loci to occur together more or less often than expected by chance. Now, this is a term we use in population genetics, so we have to measure it in a population, not in a family, and it often varies in different populations. Remember, during meiosis, chromosomes are independently separate. So then alleles on different chromosomes can be assumed to separate independently too. Now, when we use the term linkage, it means that there are two on the same chromosome. Linkage equilibrium means they are linked or on the same chromosome, but if the chance or frequency of getting each allele is the same, then they're in equilibrium. Almost like they're randomly independently separated, or even on different chromosomes. Linkage disequilibrium means it seems like something's off, Like the frequency of them happening together is a lot more or a lot less than by chance. So in school, have you ever been randomly assigned into pairs to do a project? So let's say there's 6 people in your group, like these people. So there's 5 different people you can work with. So let's say your teacher draws out 2 names at a time out of a bowl every day. And on Monday, she picks you and Mark's name. And on Tuesday, she picks you and Alice. And on Wednesday, she picks you and Alex. And so on and so forth. And it's different every time. But what if, instead of that, every single day, your teacher pulled out your name and Mark's name together? Everyone would get the sense that there isn't a same fair chance of working with you. There's a disequilibrium because it seems like you're linked to Mark, like your names in the bowl are glued together. Okay, the next term we have is mosaicism. The definition is that there is a presence of genetically distinct cell lines in the same individual. And this can take the form of somatic mosaicism, in which the mutation arises from mitotic errors after fertilization and propagates through multiple tissues or organs, or gonadal mosaicism, when the mutation is only in egg or sperm cells. Now, if parents and relatives do not have the disease, you can suspect gonadal or germline mosaicism. A good example of mosaicism is McCune-Albright syndrome, which is due to a GS protein activating mutation. It presents with unilateral cafe au lait spots with ragged edges, like you can see in this image, polyostotic fibrous dysplasia, which is when bone is replaced by collagen and fibroblasts, and at least one endocrinopathy, like precocious puberty. Now, this is lethal if the mutation occurs before fertilization because it'll affect all cells, but it's survivable in patients with mosaicism. Next, we have locus heterogeneity, which means that mutations at different loci can produce a similar phenotype, and one example of this is albinism. How many different causes of albinism can you think of? The diseases or conditions that you're naming or thinking of right now are unlikely on the same locus or even on the same chromosome. Next, we have allelic heterogeneity, which means that different mutations at the same locus can produce the same phenotype. And one example of this is beta thalassemia. There are different point mutations in splice sites and promoter sequences on chromosome 11 that can all lead to decreased beta globin synthesis and beta thalassemia. Next, we have heteroplasmy, which means there's a presence of both normal and mutated MT or mitochondrial DNA, which results in variable expression in mitochondrially inherited diseases. An example are all the disorders in which mitochondrial DNA is passed from the mother to all her children. Can you name a disease of mitochondrial inheritance? One of them is MELAS syndrome, which stands for mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. And lastly, we're going to discuss uniparental disomy. Uniparental disomy is when offspring receives 2 copies of a chromosome from one parent and no copies from the other parent. This can be for 2 different reasons. If it was an error in meiosis 1, it's known as heterodisomy and heterozygote. It is known as isodisomy and homozygous because of either a meiosis 2 error or postzygotic chromosomal duplication of one of a pair of chromosomes and loss of the other of the original pair. Now, there are some considerations. So even though we're getting 2 copies of a chromosome from the same parent, uniparental disomy is still euploid because it has the correct number of overall chromosomes. We never got a copy from the other parent. Also, most occurrences of uniparental disomy result in a normal phenotype. We can specifically consider isodisomy in an individual manifesting a recessive disorder when only one parent is a carrier. Now, 2 examples are Prader-Willi syndrome and Angelman syndrome. Can you remember what type of genetic disorders these are? You're right if you're thinking of imprinting. Now remember, in Prader-Willi and Angelman syndrome, one parental copy is silenced by methylation, and the syndrome occurs when the copy that isn't silenced is deleted or mutated. They could also occur if the offspring receives two copies of the silent chromosome, as in uniparental disomy. Okay, let's review a couple definitions. What is the genetic term for when not all individuals with a mutant genotype show the mutant phenotype? Yep, great job if you said incomplete penetrance. Let's do another one. What is the genetic term for when both alleles contribute to the phenotype of the heterozygote? Yep, that's codominance. Now let's revisit our case. She has the BRCA1 gene mutation. Is there any chance that she will not develop breast or ovarian cancer? There is some chance that she won't. Now, how come? Because again, BRCA1 gene mutations do not always result in breast or ovarian cancer because of which genetic term? Yep, incomplete penetrance. Not all individuals with a mutant genotype show the mutant phenotype. The risk of expressing the phenotype is based on percent penetrance and the possibility or probability of inheriting the genotype. So, the bottom line here is that it's important to understand terminology relevant to genetics, especially in association with clinically relevant examples. Well, that's all I have for you here. Please give this video a thumbs up if you liked it, and it'll give you the opportunity to submit a comment if you have any feedback or questions. Thanks for joining me. Study hard.